Hello students, welcome again for biology lecture. Last time in the last lecture we have discussed about what are the things necessary for protein synthesis and so many things we have discussed so far that ATP is required, then activation of various types of amino acids that is required, then the tRNA it should be joined with the tRNA to form the uh, amino acid tRNA complex and various types of the enzymes and different things which are necessary for protein synthesis. So we have discussed so far. Now let us start with the actual mechanism of protein synthesis. So let us see. This involves three steps. There are three steps in the mechanism of translation, and here it is the initiation, first process. Second is the elongation, and third one is the termination or end of the process. So let us see the PPT through which we can observe the things how they are present and what the necessary things that occur in this. Okay, so let us see. Okay, so this is all about the mechanism of translation, that is the synthesis of polypeptide chain. So these are the three steps what I told before, that is the initiation, actual the process of initiation, second is the elongation, and then the termination. So that ends the protein synthesis process. Now let us discuss about the first one, what is initiation of polypeptide chain, that is the starting of protein synthesis. So here, the first and most important thing for the initiation, that is the activation of amino acid is essential. So activation of amino acid takes place with the help of energy, and that energy is obtained by ATP, that is the adenosine triphosphate. So it is a must and it is a needful for the activation of amino acid. Then the next thing is that small subunit of ribosomes attached at 5 end. You can see here at this end, this is the mRNA, and this is a subunit of ribosome RNA, that is a ribosome. So it is a subunit, small unit attached to the mRNA. Then next one, this attachment is towards the phi end. That means it is initiation. At the phi end, it is the starting of the protein synthesis process. So it should be joined with the phi end. So the first joining of this at the phi end of mRNA. Then this first initiation codon, as we have discussed that AUG that act as a starting codon, and it initiates the process and it binds with the tRNA bind with AUG codon. So transfer RNA first it binds with this small unit and tRNA bind with the AUG, because on tRNA we must know that this is tRNA. So this tRNA having the anticodon, so it recognize uh, the complementary nitrogen bases which are written on the mRNA. For example, if it is a AUG is there on the codon or the phi region, phi end of the mRNA, then on the anticodon, what should be there? It is a AUG on mRNA. Then here it is A always pairs with the U, that is the uracil, because we know that instead of thymine in RNA, it is replaced by uracil. So U is there, A that join with the U as a complementary nitrogen basis. Then U always again join with the A. So U, A, and G that having the complementary with the C. So this is a having UAC, which is present on the anticodon region of tRNA. Okay. So in this way, the binding process takes place as the complementary nitrogen bases with mRNA and tRNA takes place. Next thing is that about after this formation, after this joining or binding process, then the larger subunit. So you can see here a large subunit is there of ribosomal RNA that joins here along with. So it joins with the small subunit which require the Mg plus plus ions. So magnesium plus plus ions are required for the joining of this larger subunit with this smaller and mRNA along with. And so in this way, this is the second step. So the first one, that is of tRNA that joins with a small subunit of ribosome join with the mRNA at 5 end. Then second process, this larger subunit join here along with the tRNA, and then the charged tRNA. So charge, that means activated. You can say it is a charged tRNA with the amino acid that occupy P site and A site is vacant. So initially A site is not utilized but directly P site. That means these there are three sites. You can see here E site, then P site and the A site. So A site from which here this ribosome join and this tRNA floats and come and join to directly P site for the first time. That means for the initiation. So this is all about the initiation process that completes. Okay. So the first one I briefly again we will discuss this in briefly. Smaller subunit join at the mRNA at 5 end. The tRNA which is activated join here, which having the complementary anticodon that join here bind with the written on the mRNA. So this is the first. In the second, larger subunit of ribosome come and it binds with the smaller one. So along with the tRNA, now it joins. And in the third one, the activated tRNA that occupy the P site. So on the this ribosomes, three sites are there, EPA. So to the P site, it directly joins along with the activated amino acid. So in this way, this is the first process, that is the initiation. Okay. Now next one. If you see here, next process. In the next process, let us see here big. Okay, so this is the elongation of polypeptide chain. Now, this next process, what happened? This activated amino acid added one by one to first amino acid. So here you can see here what the elongation, how it proceed. The first one that which is present to the P site. Okay. Now the second amino acid, which is again having the same process that is activated, it should be activated and having the amino acid. So this amino acid is activated as ATP and binds with the tRNA amino complex. So second tRNA that is again having the same type of the condition that it should be with activated and it forms tRNA amino acid complex. So it joins with the or enter, it comes and enter in the A site. Already the first amino acid having with the tRNA complex and it joins. Now you can see here the amino acid of the first one. Of course, it is a methionine, and this is the second one that is a amino acid that joins with the first one. So in this way, it is by polypeptide chain. So the joining or the binding takes place, and then this next process what happened? The codon recognize, that is the recognition. The codon recognition amino acid complex enter at A site. So because of this, you can see here this site where the amino acid having sorry that tRNA having anticodons. So it recognize the codon which are written on the mRNA. So the first thing, the same thing that is happen and it repeat with the next tRNA complex. It recognized by this anticodon and it join with this codon which are written on the messenger RNA. So this joining takes place, and then next what happen? Amino acid at P site. So this is at P site amino acid, and amino acid on tRNA join the peptide here, peptide bond That is because of the enzyme, that is a ribozyme, which act as a catalyst. So it act as a catalyst, and first tRNA at P site. Now it is kicked off. That means it goes to the A site. Sorry, E site. So in this way, this after the joining or formation of peptide bond between the first amino acid, that is brought by first tRNA, it join with the polypeptide bond with the another amino acid that is brought by the second tRNA. Okay, so in this way this process takes place, and now this vacant tRNA, now this goes to the A site. So translocation of tRNA moves from A site to P site. Translocation, that means the movement of this total structure along with the ribosome, this takes place. So tRNA moves from A site to P site. This is this A site, and then it moves to the P site. So both subunits move along in this direction, so along with the tRNA and mRNA, and in this direction it goes. That means the 5-3 direction it moves along with all this unit, and then this mRNA, uh, that that remains as in the same position, but ribosome moves from this 5 to 3 position, and uncharged tRNA, now this tRNA, that is discharged, and it goes to the E site. So this is the E site, from P to E site it goes, and the process repeat, and amino acid is added to polypeptide process occur in a very short period. So it is very in a short period, this process takes place, less than zero point 0.1 second. This process is very fast. It is a zero point 0.1 second in a fraction, you can say. It joining with this polypeptide chain, and the third tRNA arrived at A site. So second tRNA discharge from E site. That means it goes to E site, and from this E site it releases, which is now vacant. That means this tRNA now, which was with amino acid, now it become vacant, unloaded. You can say unloaded, and then this tRNA floats from this E site, exit site. It floats, and now it become unloaded, vacant, and it goes in the cytoplasm to bring another type of the amino acid. So in this way, such process takes place, which is called as the elongation process. Here you can see here the first one that join with the second one, and then second joins with the third one. In this way, this process is known as the translocation. Translocation, where the ribosome, that as a unit, smaller as well as bigger unit, that moves from 5 to 3 site. So first 3, then second 3, then again next 3, like this. So the movement is all in it. Along with this, when the polypeptide chain, that is the bonding takes place between the amino acid, then this tRNA that moves to the P first, it arrives, it goes to A, then it moves to the P, and then after the uh, vacant or after the unloading of the amino acid, it releases with the E site, and it floats again to bring the another type of amino acid. So, but it brings the amino acid which should be specific type. So in this way, such a type of the arrangement is there, where from 5 to 3 direction ribosomes move. It is a translocation, and tRNA that moves from A to E, and from E it is exit. E side that represent the exit, and this formation of polypeptide chain is there. So one by one the process repeat, same process that is a repeat, but amino acids are different, and so that the tRNA are also different, because they bring the another amino acid, and as per the message given, so it is totally controlled by the mRNA codon. As per the codon only, this tRNA can be recognized as a complementary nitrogen bases, and in this way such process repeat again and again, and then it forms the long chain of polypeptide. So it is called as a polypeptide chain, and then afterwards the next is the last termination and release of polypeptide. So what happens here, At this stage, when it goes to the another end, that is the 3 end, that is the termination codon to the mRNA. Now the process stops because for the last 3 codons, you can see here 3 or 1 or 2, it may be, it which is known as the termination codon, does not specify any type of the amino acid, but it is a vacant. And this 3 end now gives the message that to stop the protein synthesis. So what happened here? The first thing is that this ribosomes first get separated from each other. mRNA, this mRNA release into the cytoplasm and it is disorganized. Or the total use of this mRNA is finished now and it is useless, so it is disorganized or dissolved, you can say, in the cytoplasm. Then the chain of this polypeptide is there, which represent a particular specific type of amino acid. So in this way, such processes takes place. But one more thing that we must know, that there is another thing we should know, that at the end of the 3 as well as the 5 end, before the methionine, that means and last at the 3 after the termination codon, there are again having some additional sequences which are not translated. They are not translated, but they are referred as necessary factors for the completion of this polypeptide chain. And hence, those are useless. We can say useless for the polypeptide chain or the amino acid, but they are required for the completion of the process. And it is called as the untranslated region. It is called as the untranslated region. So it is a UTR, untranslated region. So this is essential because for the completion of the process of polypeptide chain or the completion of protein synthesis process. So at the both the ends they are present. That means the UTR present at the 5 end as well as the 3 end. So these are essential for the completion of the process. That is the efficient translation process it requires. In this way, so this mRNA is also released in the cytoplasm and it gets denatured by nucleases immediately. That means immediately it is destroyed because of the enzyme activity. It is called as the nucleases enzyme. And this mRNA now, uh, as its lifespan is very short. Why? Because only the function is given to this mRNA, that is the protein synthesis. And because of this, the function over, that is the protein synthesis process over, then the work of this mRNA is also over. So in this way, such a type of the mRNA lifespan is very short. So it is the short lived. And in this way, this total process of the protein synthesis that over. So this is a very interesting thing, that this process takes place a very short time, less time, and it follows all this in a very short period. That is starting of this uh, process of protein synthesis, which started with the initiation of polypeptide chain, and then the elongation of the polypeptide chain, that is the protein, and then the termination and release of polypeptide. So this ribosomes, two units get separated from each other, and this polypeptide now chain, it act as a protein. So it is a protein synthesis. So here you can see here the polypeptide chain is there, that is nothing but the protein, and which is required for the cell, which is required for the living organism. So as per the need, it may be of the plant or it may be of the animal, these proteins are synthesized and it is totally controlled by DNA. It is totally governed by the DNA. And as per the need, these proteins are synthesized. And the process is very short. That means within the fractions of second, this process, binding process takes place. And this total process of protein synthesis require very less time. In this way, this is all about the protein synthesis process in our body, especially in the cell. And various types of the enzymes are involved. Then the energy required, then raw materials, as we discuss about this. Raw materials are mRNA, ribosomes with these two units, small, larger unit, then tRNA, uh, that is having the energy source, ATP, adenosine uh, triphosphate as an energy source, then the activation of amino acid with the tRNA to form the tRNA complex, amino acid complex, and the different enzymes what you discuss further, that is so many enzymes are there. Those are not given in detail in our syllabus, so just remember that some enzymes are needed and which play an important role during the protein synthesis. So in this way, the process of protein synthesis that occur and takes place in each and every living organisms. So for each body, each living organism, proteins are different. As we know that plant proteins are different, animal proteins are different. So in this way, these proteins are synthesized as per the need of that body, of that cell, and as this all information that is given uh, by the DNA, that is totally controlled and governed, and the message is given on the mRNA. So mRNA, rRNA, and tRNA all are necessary for protein synthesis. So without this, the process of protein synthesis cannot occur. So let us stop here, and we will meet again for the next lecture and the completion of this chapter. Okay.